Okay, good morning, everyone. I hope that you can hear me all right. It's Matteo here speaking from Pisa, Scuola Superiore Sant'Anna, and I'm here to introduce you the workshop for Work Package 1 activities within CAPITALISE. So people are still logging in, but I'm mistaken, so we will move ahead and start the presentation. First of all, I want to thank you for attending. I'm very excited to be here. We have a nice lineup of speakers. We're going to speak about some of the preliminary findings of the project, but we will try to do that keeping general overview about the mapping methods that we are using with different germplasm collections. So we'll try to make something which is interesting both for people from within the consortium, but also from people who might join from outside, to whom my special thanks go. So, without further ado, we are on GoToMeeting platform, so I hope that everything works well for you. If it doesn't, please write in the chat. You should be able to access the chat, and this is one of the tools that you can use to make a question to the presenters. The other way you can do that, it's by raising your hand, and hopefully I will see your raised hand, and I will open your mic and let you interact with the speaker. Okay, so let's start from the beginning. So we're here within the CAPITALISE project. This is a Horizon 2020 project which started in 2020, which is contributed by 19 organizations from 8 different countries. We have partners from all over Europe, Israel, and Ethiopia. And in a nutshell, the project is aimed at leveraging and exploiting natural variation to improve photosynthetic efficiency in 3 key crops, which are maize, barley, and tomato, and you see picture there. Within the consortium, we have a very broad, I would say, representation of different expertise going from crop physiology, molecular biology, genetics, phenomics, and so on. And so as a team, we try to connect the dots, and to join forces to support the characterization of these allelic variation and hopefully connect it to phenotypic traits which can improve European agriculture in the years to come. Work Package 1, it's the work package within CAPITALISE which is presenting this workshop. And Work Package 1 deals with the management of the germplasm collection in the project, deals with the genomic characterization of these germplasm resources, and overviews the forward genetic approaches which are used to identify Qtls and GWAS, to perform Qtl mapping and GWAS on the panels of diversity. So this is basically what you will hear about today on different crops, and I will shortly come to the program that we prepared for you. But so what is the reason for our Work Package 1 workshop? The reason for that is that we are at a time now in the project when we start to gather information and preliminary results, not only about the genetic diversity of the collection that we are using, but also in the phenotypic diversity, specifically in relation with photosynthetic efficiency traits, which are measured in other work packages of the project. And of course, we want to use this opportunity to catch up with the preliminary results from GWAS and Qtl mapping from the project, but also to display some mapping approaches to forward genetics that we are using. So the presentation that follow will try to make a good mix of some delivering of results, but also showcasing methods which are used to mapping in different populations which are used in the project. And of course, this is something that we are really excited to show you, and especially also to the people which are outside of the consortium, but also, of course, inside the consortium, we want to map know-how and connect results and expertise across crops and beyond. So I really encourage you to interact with the speaker, and we devoted plenty of time during the day to have Q&A sessions to dig into these themes. So the program for today is contributed by researchers from Sant'Anna, which is the university where I come from and where Leonardo comes from. the Agricultural Research Organization of the Volcanic Center in Israel will speak you about barley, and the Hebrew University in Jerusalem will tell you something about tomato used in the project. And so we have a lineup of young and excellent speakers, and I really look forward to hearing from all of them. I have the duty to introduce the meeting. So besides making this a very quick introduction to Capitalize, I will now go on and show you, try to wrap up what is what are the topics of research that we are following through within this project to try and improve agriculture. So bear with me, it's going to be a very general presentation, but I hope it's going to be useful also for the people who are not quantitative genetics by training. So I don't need to convince you that modern agriculture is facing a lot of challenges. We have climate change, which is on the rise. What you see on the bottom right is a GIF, which is not starting, but if it would start, would show you the rise of temperatures across different months in the world, which is something which is quite alarming, but I'm sure that you are all aware of that. And of course, this means increased frequency of extreme events like heat and drought, emerging diseases, extreme climatic events like flooding, and of course, modern agriculture must deal with this by increasing as well the sustainability and addressing the increased food demand that comes from emerging countries. And so in order to do that, well, this is a very momentous challenge, of course, but the kind of perspective that genetics can bring to the table, at least the way I see it, is that we aim to identify and manipulate genetic factors to speed up the production of new varieties. So the idea is to use the knowledge that we are developing also within Capitalize, but of course in many other projects and in many other scientific endeavors, to source into natural diversity, natural allelic diversity, and try to use this information to support the production of new varieties which address these challenges, including a higher efficiency for photosynthetic traits, of course. The way we see that is that there are three pillars to this endeavor. The first one is diversity or agrobiodiversity. The second one is genomics, and the third one is genetics. By combining the information coming from these three pillars, we can make advancements towards the acceleration of the production of new varieties of breeding relevance. And my presentation is built up around these three pillars to see and try to discuss how these are connected towards the ends of our endeavor. So the first pillar is diversity. And what you see here, this is actually not related to the project, but is related to the activity in our research group. This is a collection of wheat varieties from a village in the highlands of Ethiopia, which shows, well, I think it's a good representation of agrobiodiversity. So this is all different spikes, which of course bear different traits, not only in terms of agronomic adaptation and agronomic desirability, but also in terms of adaptation to local climates. And so this is the kind of diversity that is the raw material that breeders shape into new and improved varieties. So tapping into agrobiodiversity and natural diversity means accessing new allele pools and new allelic combination of potential relevance of breeding. So without this diversity, without natural variation, without agrobiodiversity, there cannot be any crop improvement. And actually, if you look back into the process of breeding, actually what we are doing today within Capitalize and beyond, it's just the continuation of an effort which started roughly 10,000 years ago with the Neolithic Revolution. And I'm putting here a couple of pictures here. This one is Göbekli Tepe. It's one of the, well, probably the oldest settlement known right now. It's in the south of Turkey near the Karkadag mountain region. It's of course associated with the domestication of wheat, and there is a lot of very compelling and very fascinating literature connecting the origin of agriculture with the origin of modern societies as we see them. And of course this is mentioned in the New World, for example in the maize Central Brazos Valley, when where maize was first domesticated. So this was thousands of years ago, but then fast forward, we get to now, or the last century. I'm putting here a picture of Roman Warlock, which is a Nobel laureate for peace, and he's the father, Nobel father of CIMMYT and modern breeding. And of course, at the turn of the 1960s, the Green Revolution, what has been called the Green Revolution, started to, thanks to improvement in genetics, in chemistry, in mechanization, really provided with a step change in the production of crops. And if the Green Revolution is here, now this is an old picture actually, but now we are towards the end of this line. But what you see here and what we are doing now, it's just a refinement of a selection process which starts from very far back in time. And so this is the way we see these projects like CAPITALISE or other projects trying to leverage this natural diversity into crop improvement. It's the legacy of this project, of course. These projects are the legacy of breeding and crop selection, which dates back to the origin of modern societies. And this process of selection, of course, has some effect in the kind of agrobiodiversity that we see today. So if we go from wild relatives where we started our domestication process, which then went through early domesticated traditional varieties, land races, breeding material, and up to elite varieties, which are now cultivated at least in the so-called industrial agriculture. Of course, there's a gradient going for increasing agronomic performance and increasing agronomic desirability, but there is an opposite trend, which is in terms of genetic potential. So what this pyramid represents is the narrowing down of this diversity, which asks us as geneticists to try and go back to materials which are in previous steps of this ladder and try to rescue some of this diversity and use this diversity to accelerate the crop improvement process. And this is something that we are doing in CAPITALISE, and you will hear about, especially when speaking about Barley with the collaborators from the Volcanic Center. So there is enormous diversity out there. And then, of course, we need the tools to access into that. So the second pillar of this modern approach to breeding, it's that of genomics. And well, this is just to show you how far we went with genomic technology in a very short time. This is what you see on the left. This is a photo of a Sanger sequencing. I'm sure that some of you will be familiar with that. And this is how sequencing was done, not in the Neolithic Revolution, but 30 or 40 years ago. This is like, radioactive staining. It's a lot of complexity to get very little information, what seems to be very little information nowadays. What you see on the right is what we are using now. And also we are using within Work Package 1 of CAPITALISE. This is a mini ion sequencing machine. It's Oxford Nanopore technology. It's basically a USB key where you can load in DNA. It's a sequencer machine which can be plugged on your laptop and produce enormous amount of information. And this kind of amount of information is shown by the plot in the middle, where you see the kind of throughput increase in the last few years thanks to the development of these genomic technologies. And of course, these genomic technologies are needed, are a staple, to address and tackle the complexity of the genomes of crops, since we're speaking about crops, but of course of any realm of living beings. And us being, engaged in crop genetics and crop improvement, we need to deal with a sheer amount of complexity. And what you see here is a plot from a paper from 2017, which shows basically the genome assembly size of different organisms as a function of repetitiveness. So the amount of what used to be called junk DNA, but it's not really junk DNA, but it's transposable elements and repeated sequences, which can all together amount to up to 60 or 70 or even more, 80% of the genome of complex plants. And you see maize is over here, sorghum is over here, wheat is out of the plot because it's huge genome size. I'm pretty sure barley is over here somewhere. And so, of course, this complexity and the repetitiveness of the genome is a function of complexity. And so, for more complex, for more repetitive genome, you need more you need specific type of sequencing, which are not really the theme of this talk. But the message here is that basically we have now a whole array of sequencing technologies, which goes from Sanger, which is what I showed you, the old school sequencing method, to Illumina machines, to PacBio, Oxford Nanopore, and everything in between, which allow us to produce genomic information and address this complexity. So, but then the question becomes, now we can basically sequence everything, and we can use sequencing as a very high throughput. So, but how can we derive the meaning of these sequences? So this is the challenge of the post-genomic era where we are in. And so the third pillar, completing the picture from agrobiodiversity to genomics, it's that of genetics. And genetics is the effort of connecting genes and genomic loci to phenotypes. There are different ways to do that. Here is a zebrafish photo, is a photo of mice. I'm trying to go beyond crops, but the reasoning is always the same. What you see here is a representation of two complementary methods to determination of gene function, and both of them are represented in the CAPITALISE project. On the one hand, we have what is called reverse genetics, which basically starts from the perturbation of a gene, so think mutants, and by observing the phenotypes, we can connect back the function of the gene which was disrupted and produce that specific phenotype. The other way around is what is called forward genetics, and this is the topic of this work package 1 workshop presentation. Starts from the other way around. So we do not start from genes, but rather we start from phenotype. So we start from variation, and then we use statistics and to try and connect the phenotypic variation to the variation at the molecular level. And of course, these are not competing methods, but rather are complementary methods. And many times, of course, what happens through forward genetics is then validated through reverse genetics and vice versa. But of course, there are challenges associated to that. Most of the traits of grading interest are quantitative and polygenic, meaning that basically, depending on the trait, the individual effect of genomic loci might be very small. And here you see a classic segregation. This is a Mendelian segregation. You have one locus, you have either homozygous one type, or homozygous the other type, or heterozygous in between. But as you sum up the number of loci controlling the trait, you see that the phenotypic classes increase in number because of the segregation, up to getting to a quantitative, expression. And this is what is called a quantitative trait locus. And so this is, of course, a challenge in terms of statistics, because it requires specific methods to be targeted to the identification of the different loci across the genome which are responsible for a trait of interest. So Qtl mapping is the procedure which is used to characterize Qtl and their location on the genome, and it's used to understand the relation between DNA, sequence, and phenotypes, to estimate the relative effect of sequences of phenotypes, so we can prioritize Qtls, which are more important or less important for breeding, but also to provide information about candidate genes. We will hear something about that, to speed up the process of producing better breeds, either by marker-assisted selection or other methods, including gene editing. So I have these 3 slides which I really like to show around. These are not my slides. Are made by Daniel Gatti, who is a researcher at the Jackson Laboratory, where I spent some time during my PhD. And I think they are very useful for people who are not familiar with quantitative genetics. So I will show them as they are. So imagine this is just to display you visually how Qtn mapping works. So assume that you have a population. You have different individuals which recombine diversity from any number of founder strains. In this case are 2, but this is not important. So what we do is we use genotyping loci and produce molecular markers which, across our collection of individuals that you see on the bottom, can type the genetic diversity that is present there. So this individual in this marker will be AA, heterozygous in the second marker, homozygous in the other marker, and so on and so forth. And so we have a matrix which tell us something about the genetic diversity. Then we can have a phenotype, let's say flowering time, or NPQ, or anything that comes to your mind. And you have different plants, and in your collection of plants, you have a certain range of variation, which is represented by your phenotype, right? So you have some plants which have very low phenotype, and some plants which are very high phenotype. And so these are the 2 sources of information that you need for Qtl mapping. And so the only thing that you need to do, really, I'm trying to oversimplify something which is actually a bit more complex than that, but just to give you the idea, it's that you need to go locus by locus of the genome, and what you see here on the x-axis is the representation of a chromosome, and what you see on the y-axis is the significance of the association between genetic variation and the phenotype. And what you need to do is to test in each location. Well, let me group the lines by their genotype. So I have the AA, AB, and BB, and I look at the phenotypic value that is present in each of these genotypes. And then I can try to fit a linear model or use other methods to see whether the effect of the allele is somehow associated with a different distribution of the phenotypic trait. And you see, as we go on and as we follow through this approach, you see that when you get to the end of the chromosome, you see that all individuals that have this specific genotype have a very low phenotype, while the other individuals have a very high phenotype. And this is represented by the curve of significance, which is displayed here below. And this is telling you that, well, here there is a Qtl. There is a statistical support for the presence of a Qtl. But again, this is a statistical endeavor, so it comes with a lot of caveats. So the identification of genetic bases of Qtl is a challenging effort, which depends on many variables, which include diversity in the mapping panel, frequency of recombination, the type of molecular characterization, the complexity of the traits, sampling size, and so on and so forth. And Qtl, the idea is that what we have in our hand when we do Qtl mapping are markers on the one hand and phenotypes on the other hand. And of course, we try to use these two to connect through Qtls, but there are other disturbing factors that come in between. So the thing is actually more complex than it might seem from the slide that I showed you. And this is the reason why we have different tools which are used to address, to perform this Qtl mapping. And so different type of populations. And I will quickly move towards the end of my talk and leave the floor to the researchers who are dealing with this specific type of population. Actually, I'm not showing here pictures coming from the project. So this is from a paper from Barley, but this is just to give you an idea of the different methods that are around. So diversity panel are something that is used to perform what is called genome-wide association studies, which basically use historical recombination in the population, in very large population, to identify association between markers through linkages equilibrium between markers and causative genes. But not only GWAS you can also use haplotype-based mapping, in which you leverage the fact that you control the pedigree of population which are artificially produced. So you cross different individuals, you produce F1s, and then you produce eventually recombinant inbred lines, which are immortalized, completely homozygous, which are a picture of different recombination of the haplotypes of the parental lines. And this method is very powerful because by this you can really connect back the variation in phenotypes to haplotypes, which are reconstructed following through the pedigree of the lines. And of course, this kind of general scheme can use for different approaches. There are different crossing schemes. You will hear towards the end of the workshop about backcross inbred lines in tomato, in which basically the effort is not to produce a balanced representation, as in recombinant inbred lines, as you see on the right of the different haplotypes of the parental lines, but rather to develop a set of lines in which you can identify and with a narrow specific position introgression of a line of choice. So this collection, like the one we are using in tomato, are used, for example, to produce inbred lines which are built by intercross of a reference line with a wild relative. And this allows, it's a very powerful tool to study the impact of wild alleles for the determination of traits of interest. But the whole idea, the same framework for different approaches. The whole idea is that we start from a phenotype, we map some significance, we detect Qtl with some significance, and we try to end up with the causative genes, which are then used to map, to advance the breeding pipeline. There is actually a third type of or broad collection of different type of genetic materials which we are also using in CAPITALISE, which are called multi-parental population. And the idea here is that you try and bridge the pros of diversity panels, so increased diversity, increased recombination, and the good parts about haplotype-based mapping population, so the reconstruction of parental haplotypes. And so there are different schemes. You will hear about the detail soon, but the idea is to bring together different parental lines into a common background so that each individual line which is used for mapping is actually a mosaic of a broad diversity representing a larger share of agrobiodiversity which is available in the target crop. And so I'm getting to the end of my presentation, and this is the whole point of my presentation, which I hope was not too basic for you, but was trying to give you an overview of the idea which is behind the strategy of CAPITALISE, of exploiting new germplasm to improve specifically trait for photosynthetic efficiency. And you see here, when these are different collections which are used in the project, and I try to plot them in a in a plot which goes which contains two gradients, one going from elite to exotical inputs, meaning breeding lines or wild relatives, and one going from forward genetics, so diversity used for Qtn mapping, or reverse genetics, meaning mutants. And today, in the presentation that follow, you will hear about a magic maize population, which is a multi-parental population of maize. Then you will hear about a multi-parental population. Well, actually, before that, you will hear about a diversity panel of wild relatives of barley, then a multi-parental population developed on barley, and then a backcross inbred line population in tomato. And with that, I finished my presentation. I think that we are on time. I will hand over the floor to Leonardo Caproni, who is a postdoc in our group and working on the magic maize population. So, Leonardo, the floor is yours.
Thank you, Matteo. So today I will talk to you about the magic mice population. Magic stands for multi-parent advanced generation intercross, and we use this population to map photosynthetic efficiency within CAPITALISE. So I also speak from Pisa. As Matteo told you, I'm part of the crop genetics group at Scuola Superiore di Sant'Anna. So here I'm showing how I organized the presentation. In the first part, I will talk about the features of the magic population, the development, something about its genetic properties, and after that we will move on with Qtl mapping. I will show you somehow which are the principles behind mapping a Qtl, and which are the possible approaches that can help us dissecting the genetic mechanism of complex traits, quantitative traits. And then we will move on with some like preliminary results from data that we are analyzing, and they are coming from this effort, from the project CAPITALISE. So first, to breed a very good population, very good mapping population, the idea is that we need to include as much genetic diversity as possible. So back in 2005, when this magic mice population was started, this project was started, let's say there was this very important paper by Liu and colleagues in which they analyzed a very important international panel of inbred lines, very important for temperate, subtropical, and tropical breeding. So as the outcome of this very important paper, there was this suggestion from the authors, this creation of some core groups to maximize the representation of diversity in terms of genetic diversity within this collection. So this was one of the starting points of the magic mice population, and so the selection was made according to the principles and the outcomes of this work. And of course, another very important feature that was considered to start creating this population was to have somehow a phenology that would help, like crossing the founder lines. Of course, when you want to breed a mapping population, especially with this kind of complex schemes, we are talking about thousands of crosses. So we want to be sure that everything works well. So Here, the magic mice population, something about the development of the population. So we got, it's an 8-way magic population. We got 8 parental lines that were crossed in a half diallelic fashion, meaning that 2-way hybrids were produced in all possible permutations. Then the 2-way hybrids were crossed to produce 4-way hybrids, and then in G3, the 4-way hybrids were crossed to produce 8-way hybrids, right? And then the progeny of 8-way hybrids was selfed for a lot of generation as to create recombinant inbred lines that are actually the mosaics of the genome of the initial founder lines. And here you see the 10 chromosomes of one individual as a sample. So the idea is that because one of the hybrid failed at some point, some crosses were recovered by introducing a 9th parent. So it's an 8-way magic, but there is also a little dosage of a 9th parent that was used to recover some crosses. And today, the magic mice population accounts over 1,600 recombinant inbred lines, and about 550 recombinant inbred lines were recently genotyped with single primer enrichment technology. So, what is single primary enrichment technology? So the idea of the magic is that some of the 8 founders, we have a lot of information on these parentals like B73, MO17, and F7. We have reference genomes for this population, so we can derive information to set up like ad hoc genotyping. So in this case, like pretending that we know some SNPs, we can design probes which are actually long primers that are targeting a certain region, and so we can genotype our target SNP. But if we are producing reads that are long enough, we may also discover SNPs that are not genotyped yet. So the idea of this genotyping approach is that we are using probes and we are targeting specific loci. And so the idea is that it's a reproducible approach. So you can target at the same time thousands and thousands of loci, okay? And this panel of probes, which are developed starting from information from the founders, which is actually what was done for our population, then can become community assets. I mean, they can be reused over and over, also for genotyping other materials. Okay, so the very important aspect, technical aspect of spread genotyping is also the fact that we genotype like SNPs that are close to each other, and this we will understand it better afterwards, will help us a lot in the downstream analysis when we are dealing with haplotypes for Qtl mapping. So talking about the features of the magic mice, here I'm showing you a PCA. The color dots, they represent here the founder lines. So the first 2 PCs here explain a very low amount of genetic variation, and the first 10 PCs actually explain less than 10%. So what does it mean? It means that in this population, apparently there is no genetic structure, right? And just by looking at the first 2 PCs, we see that we can really distinguish very well the founder lines. And this is a PCA based on over 140,000 SNP markers that were generated with SPECT. And another very interesting genetic feature of this population is that along the 10 chromosomes, population-wise, we observe that linkage decays quite fast. So from, let's say, around 1.2 megabases and 4 megabases. So this means that actually this population is a resource. It's a resource to study the genetic control of complex quantitative traits. So how we can deal with quantitative traits? Well, so low genetic structure, very fast decay of linkage disequilibrium. So the first thing we can do is association studies, right? So here I'm showing you a Manhattan plot for those who are not familiar with this kind of representation. Here we see all the chromosomes. We see all our SNP markers we are testing with our regression linear model or mixed linear model, general linear model, whatsoever. So the idea is that you're testing the phenotype versus the variation of the molecular information. And here it pops up a signal, meaning that on chromosome 8 at this physical position, we have a significant signal. And so these are just to show you that here are some sample data from the campaign from the 5th of Cambridge in 2021, and we see that this signal on chromosome 8, which we also got from previous experiment as a benchmark to say that everything is going extremely well. And however, with genome-wide association approaches, we are still ignoring the pedigree. So this is the power of this kind of populations. So we may want to exploit the potential of this population, of the magic population, using another approach, which is actually Qtl mapping. So with Qtl mapping, we can test the co-inheritance of parental haplotypes. So we are not testing like SNPs, biallelic markers, but we want to work with parental haplotypes, and we want to test the co-inheritance with traits we measure. And with Qtl mapping, what we can do that helps us a lot in the downstream analysis, and also to dissect the mechanism, the genetic mechanism behind a complex trait, is that we can estimate the local effect of funder haplotypes at each Qtl. And to do so within CAPITALISE, we have been working on integrating a pipeline to perform Qtl mapping, and we used this package. Our package is Qtl2, which was developed by Carl Broman quite recently. As well, we rely on Qtl2, but as well as on many other packages. So here, the principle behind Qtl mapping. So to perform Qtl mapping, what we need genotypes. So we need a genetic characterization of our recombinant inbred lines and our funders. We need a genetic map, which is not a physical map, but it's actually a measure population-wise of the recombination that happens along the chromosome with our population. Then we need phenotypes and covariates to test the association in our model, and also we need a physical map to place our association into like a physical position on our chromosomes. So the first task of Qtl mapping is to build and to calculate genotype probabilities. So the idea is that we want to, along the chromosomes of our reals, we want to reconstruct probabilistically the mosaics we were talking about. So in order to do so, here I'm showing you this cube that actually represents a bit the complexity of the data, but the idea behind this hidden Markov model that does the job actually is that at each position, so at each position on the genome for each individual reel, we assign the probability that that marker comes from funder A, B, C, or D, or whatsoever. And we repeat this like process for every position, for every reel, for every chromosome. So actually, this is how what is the outcome of this process. So in this panel, you see like how the genome of one single reel, 1220, looks like. So each panel is a chromosome. On the x-axis, you have like the physical position, and here you have all the 9 parentals. So say chromosome 9, the first like portion of this reel comes from the parent B, and the second portion from the parent D, and then again H and D again. And here, this is the outcome of our probabilistic reconstruction of the haplotypes of each reel that are inherited from the funder lines. So moving on, so now we are ready to perform a genome scan. To perform a genome scan, it's actually a good practice to include in the model. You can use many models. A good practice is to use a linear mixed model that takes into account a kinship matrix, which is actually, let's say, a representation of the cryptic relatedness. So let's say that in our sample, in our population, we might have some divergent groups for some phenotypes. So SNPs on different chromosomes may be correlated. So we want to avoid this kind of correlation. So we use kinship in our linear mixed model, but you can use also much more simple regression like Haley-Knott regressions or this kind of approaches. So the idea is that we perform this genome scan with this linear regression, and we use kinship to test, and this is the output. So here on the y-axis, you see the LOD score, which is logarithm of odds, which is actually a measure of the significant association with our phenotypes along each of the chromosomes. So now what we want to do is to calculate a significance threshold to understand which portion are significant. And we want also to define our confidence interval for our Qtl. So we perform a permutation-based significant test. So we actually take the 99th percentile of our distribution of our permuted LOD scores. We set our threshold, and with bias credible interval procedure, we determine the confidence interval of our Qtl. So what we can do now, once we have our Qtl, so here it's a portion of a Qtl on chromosome 10. These are actual data from CAPITALISE. Here we can see like we are working in 6 megabases, so a little portion of chromosome 10 at the end, and we see this peak, and we see that one of the parents has very negative effect. Is F7. You see as negative effect on the phenotype. So it means that if a allele brings the haplotype of F7, there is a significantly negative effect on the phenotype, right? So, but once we identify our Qtls, our peaks, our confidence interval, and we have our founder effect, now the power of this population is that we can project data of our founders at the Qtl, meaning that we can use the haplotypes and project whole genome sequencing data from our founder lines, and we can test variants, SNP, indels, or even structural variation. But we also can use RNA sequencing data, so they project the expression profiling of our founder lines. Maybe at different developmental stages is what we're trying to do in CAPITALISE. And so as we go inside like a Qtl, we can identify SNP in a Qtl. So if we project like information coming from the founders, maybe whole genome sequencing data, we're not testing anymore hundreds of thousands of markers, but maybe millions of markers. We can literally grab the markers from the founders at this position and then look at the association, perform association mapping within the Qtl, and maybe look if there is any causal variant within the Qtl. And what we can do in this complementary approach, we can also look at differential expression within the Qtl. So here is an example. Here we have another Qtl, another phenotype coming from CAPITALISE. So here we have this Qtl spanning, let's say, around 8 megabases. Okay, and so here we have the effects at the Qtl of the founder haplotypes. What we can do is that we can actually cluster these coefficients at the Qtl, and we grab the gene models within the Qtl, and we can actually compare differential expression by testing, for example, the high effect founder group against the low effect founder group. We can define significance and find and look and test if there is any differentially expressed gene within the Qtl. That would be a good indication to understand if we can find a good candidate following this way. So now, going back to CAPITALISE, At the moment, we are analyzing, following the process I showed you before, data coming from the work package 2. So the collection is being characterized at IPK-11 in this amazing high throughput phenotyping platform, producing hundreds of phenotypes at many time points. We are talking about more than 500 phenotypes at 25 daily time points, but also weekly time points for other traits. And so here we have a very high temporal resolution data for photosynthetic efficiency. On the other hand, we also have data coming from work package 3, data collected in field conditions from the University of Cambridge. And in this case, we have very high resolution measurements. So we have two complementary approaches we are testing with our population. So these are some preliminary results I wanted to show you. So here we have non-photochemical quenching, that is actually a mechanism employed by plants and PQ to protect themselves from adverse effect of high light intensity. And so here we are somehow looking at associations within our panel of recombinant Inbred lines. And here we identify a Qtl. We look at the founder effect. Is actually the same graph I showed you before here. So if we look here, we found 132 gene models. So we test like the differential expression between the founder haplotype having positive effect and the ones having negative, and we only find a single gene that is differentially expressed. And actually, this gene, this is only an example, has a very relevant predicted function for non-photochemical quenching. So this is an example as a benchmark of our like protocol, and see, I mean, if we can interpret like straightforward the data coming from the phenotyping platforms. And same, another example, which I think is really very relevant for this presentation, is that we're now analyzing data coming from field trials in Cambridge and from the phenotyping facility at IPK. Here we are comparing two very similar phenotypes that are measuring chlorophyll fluorescence, and we got a signal on chromosome 10, and we got the same signals at different time points also from the high throughput phenotyping platform, so from both experiments. So here we are also observing that similar phenotypes underline same Qtls in both experiments and at different physiological stages. So it seems that some Qtls are quite stable. So to make a very long story short, what I'm saying is that from our preliminary results, we are observing and finding thousands of Qtls that overlaps across time points from data coming from IPK. Of course, when I say thousands, there is still high redundancy because we are testing each time point independently. So what we want to do now is to summarize a bit these results and look at the dynamics of the Qtl along like the duration of the experiment. And from data of the University of Cambridge, we are observing about 30 quite solid Qtls, and with some matching the results of PP2. So data looks promising at this stage. But what is coming now is our perspective. What we are going to do soon is to sequence with long read sequencing our founders. We have a PhD student, Ettore, that is taking care of this process now. And what we want to do is to refine our Qtls to summarize the results, and then we want to select some materials for the second stage of phenotyping that will be made in CAPITALISE again in both Cambridge and IPK. And this summer we want to actually produce some crosses to follow some segregation of some Qtls we believe are already solid and important, and we would like to follow.
And so we are waiting for additional phenotyping season in Cambridge this year, and at the end of the year at IPK Gatesleben. With this, I want to thank the group in Sant'Anna, Maria Ricopè, Matteo Dell'Acqua, Vanne Kromdik and John Ferguson from the University of Cambridge, and Thomas Altman and Mark Auerman from IPK for sharing data, sharing their ideas, sharing their experience. And of course, a big thank to the CAPITALISE Consortium, and I thank you all for your attention.
Okay, thank you so much, Leonardo, for your presentation. Well, I'm biased, but these are interesting preliminary results. I wonder if anybody on the call has questions. You can type them in the chat box or raise your hand and I will open up your mic. Well, while people think about it, let me ask you a question. You mentioned that you are prioritizing materials for crossing. Can you tell us something more about that?
Yes, thank you very much for the question. It's actually a very good question. So in the last, let's say, months, we have been working closely together with the teams of IPK and Cambridge. So the idea is that we identified some matching Qtls coming from both experiments. So we believe that these Qtls are stable across many experimental conditions. So what we want to do is basically we, considering the founder effect at the Qtl, we want to cross different materials, bringing haplotypes that produce different effect at the Qtl. So the idea is that we want to observe what happens when we have an heterozygous condition at the Qtl, but also we want to see, look at the progenies of these crosses we want to produce, and we would like to follow the segregation of these Qtls, and of course look at the phenotypic characterization, the behavior of these materials. So this is what we are trying to do at the moment. Thanks, Matteo.
Okay, I think there's a question from Thomas Altman. I'll try to open up your.
All right. Yeah. Hi, Matteo, and hi, Leonardo. Thank you very much for your great presentation. I just wondered whether we could go targeted for certain regions and select more recombinants from the 1600 wills. You said you have 1600s from the total magic population. However, only 550 you have genotype. But now, couldn't we go back to the 1600 and to some targeted genotyping to find some or have some recombination breakpoints and so dissect more the Qtl and narrow down the region of the interesting genes and thereby limit the number of candidates?
Okay, thank you for the question, Thomas. I will take this question. This is actually extremely interesting. And yes, the idea is that the panel we are using in CAPITALISE it's actually a panel of 350 for the phenotyping at IPK and 320 in Cambridge. So the 320 are shared between the two experiments. So what we can do for the moment, we go back to the 550 for which we have genotyping data. Actually, we may also go back to the 1600, but to do so, of course, we need another effort in terms of genotyping. So if we want to find like promising materials, like some lines that bring some interesting recombination at the Qtl, well, we need to genotype these materials and then look at it. But it's only a matter of producing more genotyping data
on more lines. And the approach we use, SPED genotyping, actually allows us to do so, because as I was mentioning, we can use again the genotyping because the panel of probes is available. It's actually available to the community. So we have high reproducibility. So we can actually go back and produce more genotyping. Yes. Thank you for the question. I hope I answered the question.
Yeah, yes, for sure you did. Just to add, it's may only be a few markers that you need to check across a large number to find more recombination breakpoints between the markers that flank the QTL.
Oh yes, of course. If we focus only on few QTLs, then we can, of course, reduce the cost and the effort in terms of genotyping. At this stage, I mean, we have so many information, so many QTLs, and still we are in the process of trying to summarize and identify a set of enlarge this set of QTLs we are focus our attention on. But yes, thanks is definitely possible, of course, and is a great approach, I think.
Great, thanks.
Thanks, Thomas.
Okay. Oh, there's a question from Mark as well. Mark, the floor is yours.
Okay, I have to unmute myself. Okay.
Yes, now we can hear you, Mark. Hi.
Perfect. Thank you, Leonardo. It is very promising to see these nice results from our data. So I just have a question, because when we analyze these kind of data on our phenotyping panel, we find temporal dynamics in the QTLs. Do you find them as well with our data? So for example, QTLs for certain traits appearing for 3, 4, 5 consecutive days and then disappearing, and other QTLs popping up? Or did you not check until now?
Okay, we are yes, thanks for the question. It's extremely relevant at this point. We are in the process of doing this kind of analysis. But for example, for the phenotype I was showing you before on, for example, on chromosome number 10, that is measuring, let's say, chlorophyll fluorescence. For that trait, we see that QTL pops up at a certain point at week 3, and then raises until week 5, and then it seems like to be stable also in the field. So these dynamics, we are still in the process of checking these dynamics, but surely this is something we observe. But yet we are in the process of analyzing the data. But preliminary checking, yes, we observe QTL dynamics, of course. Yes. Thanks for the question. Thanks.
Perfect. Looking forward to seeing the final results.
Yes.
So I will ask Christina to move the presenter role to Manas Ranjan Prostri from the Arrow Study Center, and we look forward to hear from you, Manas, about your ongoing research in CAPITALISE.
Thank you, Matteo, for giving me the floor to explain our work on the CAPITALISE project. So I'm going to today explain the trait to candidate gene by using association mapping platform, how we can reach from the trait to the gene. So I will share some of the preliminary results of V1K population, which is used in the CAPITALISE, and using some of the traits that we are working on, like the circadian clock traits, and also some of the traits shared from the PSI Research Center. Also, both of them I'm going to give a brief results of our preliminary results. So just going to the yeah. So in every germplasm, there are many different novel traits. Here I give an example of rice germplasm, because I worked on rice in my PhD, and now I'm doing postdoc on barley. So I mean to say I want to say there are a lot of novel traits in every germplasm. Here, the rice germplasm, this is a wild rice germplasm, which has been divided to different gene pools: primary gene pool, secondary gene pool, and tertiary gene pool. And this has been divided to different gene pool based on their sequence similarity to the cultivated genome. The more closest are AA genome, and the one which is little out there in secondary gene pool, and the extreme one in the tertiary gene pool. As you can see, they have a different morphology, and also they have a broad genetic diversity. They are having diploid genome, they are having tetraploid genome, and they have many novel traits like salt tolerance. Some of the rice species have C4 characters, and herbicide tolerance, flooding resistance, and disease resistance, and they have also many abiotic stress tolerant characters. But now the question is how we can utilize this trait for the rice improvement or for the crop improvement, and how we can utilize the specific marker for climate resilient agriculture, and to look into the gene that are responsible for providing these factors of resistance. So yes, the question is yes. The answer is yes, we can do it. We need two things mostly. We need to have a phenotyping platform for a particular trait of interest, and at the same time, we should have the genotype or the genome marker information for all the species. And this is how we proceed by either Qtl mapping approach or by association mapping approach to find the gene responsible for the trait. So here, this is Qtl mapping and association mapping platform. This is just an overview of that one. So in Qtl mapping, which is the traditional, which is biparental mapping, we have some donor lines, we have some recipients, someone is tolerant, someone is susceptible, or then we make crosses. We create a mapping population such as reels, recombinant inbred lines, or near isogenic lines, or double haploid lines, where we design marker, we develop marker for each of the parent, we create a linkage map, and on the linkage map, we associate the phenotype with different Qtl mapping tools, and to know where the genomic region responsible for that trait. But there are many traits like those are species where this will be a little bit harder because they're perennial, and so it will be very hard to develop mapping population so fast and to do the Qtl mapping on that one. And secondly, in this approach, when we do Qtl mapping, we land on a large chromosomal region where the Qtl intervals will be quite large, and that can contain many genes. And when we use this Qtl for improvement, it can lead to genetic drag. Some undesired character can also come during this process. So there is a need to do fine mapping for sure. We need to do fine mapping to further narrow down the causal variant for the allele for that trait. And one of the also the limitation is the size of the Qtl, as I mentioned. But in case of if you go for association mapping, where we don't need to create any mapping population, rather we can use the germplasm or breeding lines or the core collection. Here, we can capture the historic or ancient recombinant events already present in those lines. So we expect that the LD will be low there, less. So it will be useful for further final map to precisely the trait of interest. So that's why we were looking for the association mapping using this diverse line. We use a lot of markers that widely distribute throughout the genome, and this is the approach. And there are also some of the approach where this is a combined approach to further make high resolution mapping. There is a combination of both breeding and also diversity line, like development of magic population and also NAM population, and this can also be used for both Qtl and association mapping. So in association mapping, we need a germplasm, and we need to have a genotyping platform and a phenotyping platform. And the model we follow for trait identification for the germplasm, we have the genotype, genome-wide scan, it's including the candidate genes, intergenic region throughout the genome, the polymorphic marker, and then to see the population structure, if there is a structure in the population, subpopulation, or different members in the population, if there is a kinship or relatedness in the population. And then we use the model for the association analysis to look for the trait, and we do the trait mapping. So we used different platforms to do this association mapping. One of the approaches we are using the Tassel platform to find our region, and also at the same time also you use some Q by E method, Qtl by environment analysis method. I use specifically this platform, the Tassel. Here you can do the upload your genotype. You can do filtering of the genotype to remove filter, and also to do imputation, and also to calculate the linkage disequilibrium or LD in the population, and we can generate the Manhattan plot to see the significant loci in the germplasm, and also we can do the genetic distance and map. So for the germplasm here, we have used two germplasm for barley. One is the B1 population, which is collected all across the different macrosites of Israel, and also we use the Heb NAM population, and which is derived from to increase the diversity in the Barkey background. There are 25 donor lines, and this population has been designed. So in this population, in particular in the hay population, there is a mixture of cultivated allele and wild allele, and this is in other way, the B1 population is completely diverse. So, and we try to map our trait in both of the population, specifically the circadian clock traits, and to see which population has better resolution in terms of narrow downing the region, and to find and to focus on specific loci in the gene. So the traits we are interested are we working, as I mentioned earlier, the circadian clock. We have a SensiPAM in our lab where we use to use this platform to generate the data. This is based on chlorophyll fluorescence, and we conduct experiment on long day and short day. We do different environments, and we continuously capture the data in every 2.5 hours, and then from the data we deduce the circadian clock traits like amplitude and period. And this platform we use for both we use all time for B1 population and B1 population line, and also for the headlines. Also at the same time, we used also field data of headlines in different condition, in high temperature, in drought condition, where we also measure yield and biomass. At the same time, also recently we received some of the data from the phenosystem of the PSI Research Center for some of the traits like area, NGDRI, quantum yield. We received many traits, but I'm going to present some of the traits because of the limitation of the time. So to go for this association mapping, initially we have to decide which marker platform or genotypic platform we should use for this B1K population. We don't have any existing, although there are existing genotypic platform, but it was not suitable for our wild barley. So we have to do some way to make our genotypic platform. That's why we take 3 different platforms where we have the data already for iSelect 7K data, then we have ExamSec data 38K around, and then we have earlier B1K genotypic genotype marker around 11K. We combine them, all of them, and we try to see all the we try to see this is covering the whole genome. I mean, this is homogeneously distributed, not restricted to some of the chromosome, and all the you can see the distribution of the markers along the 7 chromosome on the right hand side. It is quite it looks like all the genomic position or genomic region in every chromosome is more or less captured in this platform. And to this, when we compile all this marker, then we send this marker for probe designing, and we do follow many filtering criteria to make sure that the marker we design is a single hit in the genome, and it's unique in the genome. So the further we the company LDC, we design the probes, and we finally whatever the marker we got, and after filtering out further for the missing and monomorphic and the multi-allelic marker, and also removing the minor allele frequency, we reached around 13.7K marker for GWAS, and this is what we used for this analysis. As I mentioned, the genotype file, when you upload to this Tassel program, we do filtering. We can filter in many ways. We can create different filtering like minor allele frequency. And here, just for example, I do imputation because during the generation of the marker, we have many missing data in the for some of the markers. So we use the imputation. You can see the marker which is we highlighted by a circle. This is missing, and using the imputation, we filled all the missing information, and we use this for the mapping. So as I mentioned, we try to look for the population structure in the Heb population and also B1K population, because the population structure is also important in for mapping. If a population is structured, that means they don't do random mating there, and there need to be some correction to the population. For B1K, we didn't see a very good population structure. Rather, we see in the Heb population, there is from the PCA plot, we do see some population structure exists in the system. Secondly, if we see the power of the germplasm for the straight mapping, on the upper hand, you can see the chromosome 1 to 7 from the Heb family, and the lower one is for the B1K. As we see, this is the LD map, the linkage disequilibrium map. Linkage disequilibrium means the non-random association of an allele to the other one, and we see in the Heb population there is a high LD. You can see the LD blocks, but in B1K we see the LD is very low, and this is only restricted to the centromeric region where there is recombination events are relatively slow. So the B1K population has a much smaller LD. So this population has a high power compared to the Heb population, that it can help us in narrow downing our region to a very small loci. So the statistical model we follow is GLM and MLM. We know in the GLM model, we use only for the SNP and the population structure. But in the mixed linear model, also in addition to this population structure, also we look for the kinship, the relatedness in the relatedness within the members from the genotypes. So we went for the MLM analysis initially for the. I will come to our clock race a little bit late, but I would like to discuss the trait from the PSI Research Center. This is we followed the MLM model to analyze the data. So this is the data from the NGDRI trait from the PSI system. In the left-hand side, you can see they have data. This data across different developmental stages of the plant from 10 days after transplantation, 20. I mean, all across. This is not from low to high, but this is that they have all the data from. If I remember correctly, from 6 to 44 days after transplantation, a lot of data. So we wanted to see using this model if there is a significant association of any trait to this NGDRI trait. We see. I just try to highlight it. You can see I make a heat map of the significant marker that resulted from the GWAS. After we conduct the GWAS, we have the p-value or the score. From the p-value, we calculate the lot score, and we for each marker we try to arrange it from the for the different days after transplantation. So and they are color coded. The list are with the red color, and as we go, the lot score go high, so the color turns to green. So here the significant markers are you can see in this Excel sheet. Here one of the interesting things is that we found one of the markers which is in chromosome 2, and the one is also highlighted. It came consistently across all the days of transplantation traits throughout the in most of the, I think in 1, 2, 3, in 3 is in 3 developmental, except 3 developmental stages, it appeared in all of the days. So this is quite interesting. So this is something to look into. What is the and what we need to further look, and we didn't look into that to see if this marker is an LD to the nearby marker, and to see what are the genes responsible in that region, and if it's not in LD, and if it's a single and it's lie directly on some candidate gene. So this is something very robust loci in the genome that really responsible for this trait. Secondly, if I want to share the second information on the quantum yield on the low light, similar, you can see the developmental stages and also the quantum yield. Here I see on chromosome 3, the one which is here. This also seems to be very consistent, and I see increasing in the lot score or increasing in the marker p-value as we as the developmental stages go on. So this is the preliminary results, and it is interesting to look into the marker, the nearby gene, and its effect. What's the things behind that one? So also in addition to this Tassel analysis, also we conduct some Qtl by environmental interaction analysis. This is done in collaboration with one of our collaborator, A.G. Yamamata from A.G. University. He do mostly Q by E interaction, and we present some of our results on that. So the Q by E interaction model is carried on with this main effect, additive main effect, SNP effect, and interaction terms. So since we conduct our experiment in a different environments, we want to see how the different alleles behave with the different environments. So here we try to summarize how it should behave if we have in the previous slide I saw that CAPITALISE 2 effects. additive main effect and all SNP effect and interaction turn. So if only both the additive and main effect, additive main effect are significant from the GWAS results, while the interaction term is not significant, then the graph looks something like this, where you can see the reference allele versus the alternative allele, and this is the allelic effect, and this is two environment, high temperature and optimal temperature. So they are this is called persistent effect. They go parallelly each other, and there is no interaction. And if the additive main effect becomes non-significant and the SNP effect and the interaction come, so if the only the SNP effect is significant, then we see convergence effect, and we do see some kind of interaction. And the highly interactive one is called the crossover effect, where there is no significance in the additive main effect. Rather, there are the two remaining SNP effect and interaction terms are significant. Then we expect to see a high interaction for this marker between the allelic state. So we carried this experiment for amplitude to find the amplitude and period, and this is particular. This is we do on B1K lines. Although we carried on Hebb lines, but I wanted to show some results from this B1K population. So here are the different effects that we discussed in the previous slide: the persistent effect, all effect, and crossover effect for amplitude, and in the right hand side for the period, this effects. So we see we have a threshold we calculated, and we see this is Manhattan plot. This is the different chromosomes across the Barley genome, and here is the p-value of all markers, and they have been arranged from low to high based on their physical position in the genome. And where we see a pop-off above the threshold line, so it's a possible possible signals that this might be contributing to this phenotype and has some effect. So we we presented all this data in Excel format, the Qtl by environment interaction analysis, where here we have all the traits. Let's say here is amplitude and for period, and we have all the p-values here for the additive main effect, additive and p by effect, and for the q by effect. And these are the effect size, marker effect size. How do they behave under different conditions? As for example, how do they behave under high light, high temperature under short day, high temperature under long day, and optimal temperature under short day and optimal temperature on long day. And what is their p-value for their respective temperature. And here we have also the minor allele frequency and also the LD map on the right hand side. So we can know if a marker came significant. We can see really if it's is in LD to the nearest marker, or it's the signal is on the marker is the signal itself without any LD to the nearest marker. So this would really help us to know if how many genes are present, or the gene is present only on one side. So this is really helpful to further to look into the effect of this marker. So I wanted to show you zooming into candidate gene. Here in the beginning we used our Hebb panel, which is a NAM population, and we found some hits for amplitude, and we named these the doc drivers of the clock, the doc 3.1 and doc 3.2, and this is based on the significant marker, and this is the doc 3.1 based on the LD size in the genome, because I explained in Hebb population the LD size is quite high. So for the same trait which we use in the Hebb panel, the same trait or the same phenotype we try to repeat for the B1K panel. And here we see, because the B1K panel has a low LD, we see the hits that we found here is very small compared to the Hebb. And whatever the significant markers came here, we found two significant markers that came within this region, and we expect the marker is so close to the Gigantea gene, which has already known to be the clock gene and has a role in the circadian clock. So it might be responsible. When you try to look on to any of the markers which can be present in Gigantea, unfortunately, we didn't have any marker in our genotype platform, which is already included for the within this region. So we have a missing information for this Gigantea gene, but the nearby marker which is existing in they appear to be significant. So I want to also mention, and the hits we get in GWAS need to be checked in the next generation or making an active population to validate the marker, because sometimes we get the hit from the statistics analysis, but it can be spurious. If we really want to use it for crop improvement or program, we need to really validate this SNP. As for example, I have shown here there is a significant signal which is for the dry weight or the biomass, and for this marker which came significant, if we look, this is from the health panel. If we want to compare for this marker which allele contributed to what extent, we see the cultivated allele is decreasing the dry weight, whereas the wild allele is increasing the biomass. So if this markers holds true, and if you want to validate this marker, we can design an F2 or make a cross, taking one of the line which has this Qtl or which has this marker and cross it to a cultivated line which is not responsive for that one. And in the segregating line, we can do genotyping by gel-based marker or any high-throughput system. And in the next generation, if we see with this marker, the phenotype also go, and so in this way we can validate our marker. So in fact, we always try to do that one. We did that one for a different loci in our genome FRP2 too. This is another locus for the crop, and we try to validate this marker in the next generation or by making any F2 population to see if the effect is there. And if there is an effect, then it's highly encouraging to look into to other molecular studies, like to do the transcript profiles for this marker, without this marker, and also to try other analysis to further narrow down the region. So with this, I would like to conclude my talk, and thank you. I would like to acknowledge all our partners involved in this project, and thank you. Bye.
Okay, thank you so much, Manas. Very nice background picture, by the way. Okay, so the discussion is open. If anybody has question, you can type on the chat or raise your hand and I will open your mic. Okay, while people think about that, maybe I will break the ice. I've seen some very interesting Qtls reported for the across different developmental stages. I was wondering, is there any pleiotropy towards traits of agronomic relevance that you're aware of? I was thinking of a Qtl which is pretty consistent on chromosome 2. Do you know whether this was already reported in literature and whether it has a role in other aspects of barley agronomic performance?
Yeah, thank you. This is a good question. When we, as I mentioned, we worked on a number of traits from that we received from the PSI Research Center, not only on quantum yield. We have quantum yield low light, quantum yield highlight, area, NP traits, NPQLS, and so after we do the analysis for each trait, we try to look for the pleiotropy. Yeah, and yes, we do see. I cannot tell you exactly the marker you showed to me, but if I look my Excel file, but we do see a lot of pleiotropy among this marker, especially for the quantum yield and NPQ, and also for the other traits. So I can, the maximum number of pleiotropy that I saw from my last analysis around markers having 4 traits. So they can control different traits of this phenotype. And to also, I would add, we have also conducted this pleiotropy for our clock traits, the circadian clock, the genetic loci that controls the circadian clock, and also we have the yield data and also the biomass. We do see also a pleiotropy between the circadian clock to that of the biomass and yield in the field. This is generated from the machine, and this is from field data, and this is highly pleiotropic.
Okay, thanks a lot. Indeed, this is what I was thinking, because in the end, of course, we are looking at crop improvement, right? So it's fantastic that you can link phenotypes that you can get in the phenotyping machine with what you see in the field.
Yes, yeah.
Any other question from the audience? Okay, so I think we can maybe move on to the next presentation, and we will say thank you, Manas, so much for your very interesting presentation. We will now move to the next presentation from Sheva, from the same. Sorry, I hope I'm not butchering your name. From the same team, but telling us something different, and something about multi-parental population, which will combine some of the wild diversity with cultivated materials. But I leave the floor to you, Sheva, and thank you for being here.
Okay, great. So welcome to my presentation about mapping cytonuclear interactions in barley. So let's jump right in. Let's first have a look at classical plant cell. So what is actually interesting is that most of the mapping studies that we were also discussing before, they focus on the nuclear genome, which is with loci that are normally Mendelian, that follow Mendelian inheritance, which we call the nucleotype. But what we know is that there are also different genomes in the plant cell. We have, on the one hand, we have the mitochondrial genome, and on the other hand, we have the chloroplast genome. And we know that both of those genomes, they maybe carry a much more limited amount of genes, but those genes, they are involved in metabolic processes. And when we talk about chloroplast, they are also involved in photosynthesis. And it was also shown that it impacts actually the phenotype. So the phenotype is not only influenced by the nucleotype, which is the factor which is normally measured in mapping studies, but it is also measured by the plasmotype. And this is maybe demonstrated by a quite nice experiment which was done recently in Arabidopsis. So what we're actually seeing here is a design where 7 different accessions were chosen in barley, and just looking for the arrow. Okay, we have 7 different accessions in Arabidopsis thaliana, sorry. And what they did using transgene technology is that for each, they took all of those accessions and they replaced each time the nucleotype and the plasmotype. So basically, they called it hybrids, and it follows kind of a diallele design. And then they measured the performance of a range of different phenotypes, and they looked at how the nucleotype performs in every one of the different plasmotype backgrounds. And we can see that the performance of the nucleotype, for example, on the efficiency of photosystem 2, this is one trait that we're analyzing, we can see that it varies strongly depending on which plasmotypic background the nucleotype is found. And we can also see that there are various like the sizes of effects. So we can, for example, see this for those first 6 plasmotypes, we can see that the effects, although they are there, we can see that the 2 nucleotypes, they vary, like the performance of the nucleotides, they vary, and sometimes even the order between them is varying. But we can see, for example, one plasmotype from the early accession that has a very strong we may say epistatic effect, that no matter which nucleotype the Arabidopsis plant is carrying, when it has the LE plasmotype, the photosystem efficiency drastically goes down. Okay, and we are of course very interested also in those interactions when it comes to crop plants, more specifically the barley plant, which we're working on in our lab. But of course, we don't have the same transgene technology where we can just replace, totally replace the nucleotypes and swap between the different plasmotypes and nucleotypes. So we basically came up with a different approach. We developed in our lab the Barley Cytonuclear multi-parent population. And this is actually a modification of the classic nested association NAM design. And what we have here, we have 10 wild founders, which are derived from the B1K collection, and they represent a range of eco-geographical parameters of Israel. And those 10 founders, they were crossed with Noga, which is a cultivated, the cultivated parent we used in this population. And what happened is first we went to the F1 stage, then it was crossed back once to Noga as a male donor. Very important to mention here, what I didn't mention before is that contrarily to the nucleotype, of course, the plasmotype is maternally derived, which means the inheritance goes through the mother plant of the cross. And this is why when we got to the backcross 1 stage, we basically, in order to get to the backcross 2 and final or pre-final stage of this population, we crossed all the plants from each subpopulation, once to Noga as a male donor and once to Noga as a female donor. And then we got to the backcross 2 stage, where we also did some QC genotyping to see that the amount or the ratio of wild DNA is approximately as we expect it to be according to the pedigree. And at the end, we went, this population went to a double haploidization process, and it yielded approximately 960 double haploid lines. And so first of all, those 960 DHs, like it is usual in a NAM population, they are subdivided into the 10 subfamilies, each one deriving from a specific, from Noga and a specific wild parent. But it also divided in half of the population carrying the Noga plasmotype, the cultivated plasmotype, and half of the population carrying the wild plasmotype, which means we have, of course, segregation of the nuclear genomes of the cultivated and the wild donors, but we also have, you may call it a segregation, although it's not exactly segregating in the classical sense, of the plasmotype. And maybe a little bit about the diversity and the composition of this population. So those are 2 analyses that were done on this population. So at the left side, you can see Chloroplast sequencing, which was done for the founders of this population. So Dr. Lalit Tiwari in our lab is a postdoc, and he has a very efficient protocol to extract chloroplastic DNA, and that's why we were able to send this to nanopore sequencing. And what we can see here is the different B1K lines that were used to build up this population. And we also have Noga here, and we can see that there are indeed polymorphism, which even leads to amino acid changes in MATK, which is a gene, like a chloroplastic gene, which has roles in splicing, in RPOC1, which has roles in RNA polymerase complex. So we can see that there is, at least on the sequence level, we already see quite an interesting variation in this population. And on the other hand, the other side, you can see the QC genotyping we did it on the backcross 2 level. Basically, those are those plants those pollen were used to do the double haploidization. And as we expect from Bacrus 2 plants, we would expect that they have a level of heterozygosity of approximately 12.5%. And you can see that the actual values of heterozygous calls that we get for when we subdivide it in each subfamily is approximately what we would really expect from the natural ratio of wild donor DNA within the population. Okay, so where are we standing? Well, or what are our maybe nearest plans using this population? So first of all, regarding the genotype, we are using low coverage whole genome sequencing for the whole population. And for the parents, we're doing also whole genome sequencing with a higher coverage though. And this higher coverage information will be used to impute more SNPs in the population where we know that the introgression exists, but where we have lower coverage. So regarding the phenotype, what we are, this population is quite, it is only propagated for the first time this year. So we're just at the start of it, but we already are nearing 3 replicates of measurements with our circadian clock output measurement machine, the Sensipan, which Manas also introduced before. You can see a picture on the right side. You can see that basically 240 plants are basically waiting to get into the chamber where the different, where pulse amplitude measurements are performed. We can derive from these different parameters regarding the circadian clock after we transform this data to get the amplitude, the period, and so on. And we also are getting preliminary data from the propagation we are doing now on height and also on days to flowering. So this is one preliminary phenotype that we have. This is the days to flowering that we measured during the propagation. And what you can see here is that it is subdivided, first of all, according to the subfamilies, according to the wild donor. And it is also, each subfamily is subdivided into plants with the cultivated and plants with the wild plasmotype. And as you can see, what is interesting here is that the plasmotype has a different impact depending on which subfamily we are looking at. It has a different impact on actual days to flowering. So for example, in the subfamily O2, we can see that the plasmotype doesn't have any effect on days to flowering. We can see that, for example, for the O3 and 42 families, there are highly significant impacts of plasmotype, on days to flowering, which also means that the diversity obviously not only exists on the sequence level, so it confirms the fact that it has also implications on the phenotype and even on very important field phenotypes. What is also interesting is if we look exactly at the same analysis, but instead of days to flowering, we look at the circadian period, which we measured at an optimal temperature with our machine, we don't see any significant don't see any family which has a significant impact visible from the plasmotype. And this is quite interesting because especially if we're looking at those two phenotypes, days to flowering on the one hand and circadian period on the other hand, it is quite interesting where we know that actually days to flowering interacts with the the decision when to flower, it does interact with the photoperiodic pathway. It is interesting to see that there is no visible correlation, at least when we look at it this way, between those traits. I mean, there may be several explanations. One explanation is that maybe plasmotype does not impact the circadian period per se, but maybe it impacts how the plant decodes the circadian period and translates it into those field phenotypes, like, for example, days to flowering. And now if we maybe look also on go back to the nucleotype a little bit. So what we can see here, this is a GWAS which was performed on circadian clock parameter on the amplitude under high temperature and under optimal temperature. So this was done with the HEP population, which was developed in Germany, and it was published about a year ago. And the two outer shells, basically, they show significant loci with significant association with amplitude under optimal and high temperatures, where we also actually discovered certain loci which are responsible, which lead, for example, to higher amplitude in the high temperatures only. So where there is some kind of interaction between genotype and temperature. When we now want to incorporate the plasmotype, of course, the HEP population is a population which was not developed while this population is not aware of the directionality of the crossing. But what we would like to do when it comes to the CHAMP population is we would like, for example, to add another shell on the outside and to show epistatic effects, for example, or additive effects or interaction effects in general that are influencing those loci which we discover for the different phenotypes that we are analyzing. And of course, we are still waiting for the genotype data, but we already, of course, thinking of which kind of models we can apply to discover this kind of interactions. And one approximation maybe which could be possible is that we can use, for example, studies or genome scans that are used in humans. For example, what we see here, this is a genome scan with a certain biomarker, which is basically measuring, which is kind of an indicator, it's a marker for indicator of liver health in humans. And what we can see here is by applying the right model, they managed to discover a certain genetic risk factor, which is only significant in the case of women, while in the case of men, this genetic risk factor does not appear to be significant. And we think that maybe if we use this kind of interaction models, it will allow us to discover maybe certain nuclear Qtls which are dependent on a certain plasmotype or which act in an epistatic way with certain plasmotypes. And of course, a very important step once we discover this kind of Qtls is to do the validation. And here the classical validation workflow is also a bit altered. So what we are used to is to, first of all, maybe cross back several times our candidate Qtl to reach a near isogenic line with our candidate Qtl, but instead of crossing it just in one direction or not being aware at all in which direction we cross it afterwards to maybe create a segregating population, we cross it back once to Noga. For example, if it is a line which is derived from the CHAMP, we want to cross it back one time to Noga as a male donor and one time to Noga as a female donor. And then basically what we get is reciprocal segregated populations where we can first of all get a good validation of our Qtl to see whether it's acting or not in a clear-cut background, and also whether we hopefully will also allow us to understand if the plasmotype has an impact on exactly this Qtl when it segregates in two different plasmotypic backgrounds. Okay, so to summarize this presentation, so the plasmotype, which is maternally derived, it influences a lot of processes in the plant, also phenotypes which are relevant for agriculture, metabolic phenotypes, and so on. We also note that those phenotypes, they're oftentimes results of an interaction between nucleotype and plasmotype. Like there is certain, like the same nucleotype may not act the same in different plasmotypes. And as mentioned, the strategy that we are using to discover those cytonuclear interactions, first of all, we develop the CHAMP, which I presented before. We acquire genotypes, we acquire a high resolution genotype, also the phenotype acquisition. Yeah, we want to design a genome scan which is aware of the cytonuclear interactions. And as mentioned in the last slide, we would like also to validate those Qtls with reciprocal segregating populations. I would like to thank Dr. Eyal Friedman, my supervisor, Dr. Eyal Bdolach, who developed the CHAMP and is responsible for acquisition of the phenotype, and without his preliminary work, all of this wouldn't be possible. Dr. Lalit Tiwari, who did the chloroplast sequencing, Dr. Manas Proshti, who basically also figured out different Qtls that I'm working on now, and collaborations. Dr. Yamamoto, he is from Japan and working with us on different statistical models. For example, that the statistical model that I would like to develop, I'm at the end of the day, I'm an experimental biologist, so he is someone who will support me in figuring out how to model, how to build those models. And Dr. Dan Koenig from UC Riverside, he is largely responsible for the genotyping of the CHAMP population, and is also acquiring some of the phenotypes. And the institutions which are supporting us and grants, the Binational Research Fund, Israel and the United States, the BARD, Saten Union, which were very supportive while developing the double haploid population, CAPITALISE, and of course, thanks to my audience, and I'd be happy to answer any questions.
Thank you so much, Shevach. It's amazing work. I'm really fascinated by it. But I will give, precedence to the audience to ask you questions, either on the chat or raising hands. I would be happy to. Yeah, I see Thomas as a question, so I will open his mic. Oh, sorry, Thomas, you can open it.
All right. Yeah, Shevach, very nice work. Excellent and very interesting aspect to check for the nuclear plastid or plastome interactions. My question is, how would you dissect the plastome factors, so the plastid encoded genes? Currently, you're considering the plastome as a whole, but of course, you would like to find out which of the plastid encoded loci or genes is responsible for that interaction with a particular nuclear factor. So how would you go about that?
Okay, so, well, I think one obvious, difficulty that we are facing here is that if we have a certain candidate polymorphisms in the plastid genome, we can't just segregate it out and backcross it and get rid of it and then verify it in a very isolated manner. This is, of course, something that that we cannot do, and this is a kind of a limit. However, what we are doing in our lab is that we are now basically propagating a tilling population. And this tilling population, the material which was used to mutagenize those plants is basically targeted especially on the chloroplast genome. So it's known to, I think it's EMS, it's targeted especially on chloroplast genomes. And these days we're basically using a kind of a DNA Sudoku method where we are now genotype, in order to genotype all of those or to discover rare mutations in those tilling chloroplast genomes. And really, like coincidentally, this week we are getting results. We did a PacBio sequencing of those of distilling of the chloroplast DNA of distilling population. We are now getting our first mutations there back after the analysis, and I hope that this will maybe give some insight, like and will help to clarify maybe some candidate genetic factors within those chloroplastic genomes. We also have the option to do TALEN, to specifically it exists in Barley the option to target by genome editing chloroplastic genes. This also could be an option once we would maybe zoom in into different genetic factors.
Wow, that's great. Excellent. I think it's quite a lot of work to generate this tilling population, since you have multiple copies per chloroplast, you have multiple chloroplasts per cell. So to get a uniform stock may take some time of propagation, right?
Yeah, I mean, we are now at the beginning of it, so we are starting to make sense of this data. Well, I mean, now we got the first results. Like first of all, we got the results from PacBio, where we basically we pooled those DNAs in the way that the bioinformation told us to pool them, and he will of course at the end make sense of and figure out which pool has which mutation, and can then code it back to which plant was added to which pool. So it's like, and we are now really getting the first mutations there, and we will now start to validate, to go back to the field, take those, or we have of course the chloroplast already DNA in the lab. We will now start to sequence and to validate those mutations. And of course, we hope that there are not too many like hidden factors we did not consider, which will hinder this analysis. But of course, the fact that there may be many different, like that the DNA may not be homogeneous between those chloroplasts, it may be a hindering factor. But I guess that's something that we will figure out.
Yeah, look forward to see that and to hear about your progress there. Thank you very much.
Thanks.
I see a hand raised by Ian. Great to hear from you, Ian.
Yeah, no, I'll just comment on that. I mean, that's we are going to the unknown to some extent. I mean, going to the chloroplast diversity, there is also one thing that wasn't mentioned. There is also redundancy, what is called NAPT. So there are, for each gene in the chloroplast, you have copies in the genome. So that's something also to consider, which is not trivial. So there may be redundancy, there may be not, we don't know. But nevertheless, at least for one gene, we were able to find a mutant in Arabidopsis, and luckily there is only one copy in Arabidopsis chloroplast. So there is a limitation, of course, but as Shevach showed, he indicated that it was shown that there is a chloroplastic effect, but nevertheless, there are not many population who consider the chloroplastic effect, and even having that on the flowering time, it's I think it's interesting by itself. But going to the molecular level, it's going to be challenging. I agree. I mean, it's not trivial.
Thomas, do you have a hand raised, or it's a legacy hand?
It's again raised. Just occurred to me, how do you distinguish the plastid interaction from mitochondrial nuclear interactions in this case?
Okay, so of course we cannot separate those two. We cannot separate those factors, but because the main focus of my study, actually is photosynthesis related traits, I have the assumption that the chloroplast is the main player in this game. But of course, that's also why I call it plasmotype. I don't call it chloroplastic DNA when I talk about it. I talk about the plasmotype as a whole. And yes, right now I don't have the option to basically play around with it, not on the population level, because then of course it's very difficult. Maybe if we can get to it at the molecular level and do some targeted analysis there, this will maybe allowed to conclude that a certain genetic factor really that is a candidate is located in the chloroplast genome. But yes, that's of course something we cannot 200% exclude and include.
Mm-hmm. Yeah, fair enough.
Another point in that, Thomas, is that historically, if you look on the literature on the variation in Barley, there is hardly variation in the mitochondria. When people reported variation in the chloroplast, and you probably know this also maybe from Neil's work now going on, most of the variation, you do see tons of variation in the chloroplast, you hardly see in the mitochondria.
Okay.
So it's possible that it's coming from there as well, but people have reported, I think it was work done by, it was also done in IPK actually, and they show that there is hardly variation in the mitochondria, but still, it's something to take into account.
Mm-hmm. And photorespiration, I think, also includes, of course, the mitochondria. But anyway, fair. Yeah, sure. Thanks.
Are there any other questions from the audience? Okay, I'm gonna take my chance and make my question that's also related to what Ian was saying. So I'm actually surprised to see how much diverse the chloroplast genome is across your collection. And I see that actually, maybe I'm mistaken, but there is one line among your parental lines which is very different, or at least has nucleotide variation at many different loci. So any reason for that?
You mean here? Do you see my screen?
I do. No, when you go to slide number 5.
Okay.
Or maybe I'm reading. Oh no, okay, I'm reading it in a wrong way. Okay, no, sorry. So the parents are the vertical one, B1K4918.
Which slide? 5?
Yeah, the slide that you are showing right now.
Okay.
I'm trying to read the table with the nucleotides substitution, and it seems that there is one parental line which is, or at least one line.
Okay, which one?
B1K4918, the last one.
Okay.
Is there any reason why? So, well, if you want to rephrase my question, is there any is the diversity that you see at the chloroplast level related in any way with the collection, the way the collection, the B1K collection was done?
So basically, the collection was done mainly, if I go back to these slides, that it represents different, geographical and also, rainfall gradients. This was more so you can see those different circles there. Those are basically the different regions in Israel which were considered to take from there the founder lines. Israel has a very broad range of different environmental conditions. We have like very arid regions, desert, but we also have a very fertile northern region. We have coastal regions. We have less salty, more salty regions. So this was more the consideration was more there than looking on the sequence level, what is more diverse, especially also, yes, this was actually the main consideration.
Yeah, but then, so my question, let me rephrase my question. So now that you have the sequence of the chloroplast, can you trace it back to specific genetic clusters on the landscape? I mean, does it have any connection with, evolutionary or adaptation paths that the different populations, the B1K population might have had?
That's a good question. I actually never looked into this. No, I mean, something that is that should be considered maybe. Yeah, it's interesting.
Okay. So I see that Lalit is also on the call. I will drop you an email because I'm very interested I think it would be interesting to do something like what you're doing on the maize population as well. And so we don't know anything about the chloroplast variation in our collection. So we will try to follow up on your steps and see whether we can derive at least the information about, the chloroplast pedigree in our population, because we can use that as a covariate in the mapping, as you showed in your last slide. But anyways,
Sure.
I don't want to bore everybody with our specific research interests. So I don't see any other questions from the audience. So then we can thank Shevach and Manas as well, so the whole Arrow team, for the wonderful science they showed us. And we can start again. You see that we are Italians by how many coffee breaks we plan in a single meeting, but I hope that they've been a bit refreshing to you. So now we are closing with Barley, and then we are moving to the presentation from Shai and from the Hebrew University of Jerusalem team about the tomato bean population. So Shai, the floor is yours, and thank you for being here.
Thanks, Matteo. I hope everyone hear me well. Hello everyone, my name is Shai. I'm a PhD student in Dani Zamir's lab from Israel. And today I'll present about Qtl analysis and Qtl mapping in tomato beans population. And actually I don't show some analysis from the CAPITALISE project, but I show how to use the R software to actually doing the Qtl mapping. So we first start with the basic for the Qtl analysis of the analysis of quantitative traits, and that is the distribution of the phenotype in the population. So when it comes to a single gene that affects the phenotype, the distribution can be separated into 3 genotype groups, as we see in the first graph. And also you can separate with some environmental effects. But in contrast, the greater the number of genes and the environment that affect the phenotype, the more continuous the distribution, and the different genotype groups cannot be separated well. So the pipeline of the genetic dissection of complex trait is that we have some genotype, some population. We make an experiment with a replication in a different environment. And then we collect the phenotypes on the population for each line, for each genotype, and send the population for genotyping to get some markers, DNA genotypic data. And then we can do an observation, some association between the phenotypic data to the genotypic data to perform a Qtl analysis that can explain us the phenotypic by the genotypic. So the principle of Qtl mapping is that Qtl mapping is based on the segregation of genes and markers by chromosome recombination during the meiosis, and that's what allowing us to the analysis in the progeny. And the partition of mapping population into different genotypic classes based on genotype marker and apply correlative statistic to determine the significant difference of one genotype with another with respect to the trait being measured. So, for example, here it's some real population, and in the green segment is the cultivated tomato allele, and in blue segment is the wild type cultivated allele. And as we see here, it's all the genotypes, the lines, the different lines that every line contains different segments of the wild type allele by recombination. And let's assume that all the lines that have the green segment, the green interval in this specific region have a big fruit, and all the lines that have a blue segment in this region have small fruit. So we can suspicious in this region that affect on the phenotype that we measure. So this is the basic of Qtl mapping analysis. So before we move on to the mapping demonstration, actually in the R software, I want to introduce you the mapping population that we will work with. And it's also used by other labs that we work in a collaboration as part of the CAPITALISE project. So in 2007, we start to develop a new build population with a new unexplored wild species from Pinella accession. It's called LA5240. Its genome is published in Cell in 2017. So what's different this wild species from the LA716 is that it's fully self-compatible. It produces a lot of seeds. So it's a really advantage in wild species. So in 2017, we crossed this Pinella to a modern cultivated determinate tomato for the processing tomato. We call him Leah. So we produce a F1 hybrid. This F1 continue into two different pathways. The one is a Becos1 back to the Leah background to create 2,000 plants, Becros 2. And then we continue with selfing until we get Becros 2 cell 5. In the another pathway, the second pathway, we take this F1 and cross him to a big fruit indeterminate tomato for the greenhouse tomato. And we backcross 1 to the top. We call him top, we backcross 1 back to the top, backcross 2, and selfing until we get backcross 2, self 5. So in the Leah background, and this is the background that we're going to do the analysis, we have 1,500 plants in the population. So it's extremely high population that every line contains different segments of introgression from the Pinella. And it's because the size of the population and the recombinants between the lines, the resolution of the population is very, very high. We measure about a recombination event every 18 kb. So it's really high. It's apply us to mapping even into genes, recombination even into genes. So based on this population, on the Leah population, on the genotype of this population, we build a set of 53 lines that cover the entire wild genome in the various introgressions. So the red one is the cultivated alleles, and the greenhouse is the segments of the Pinella allele. So you see here that in each chromosome there is a different line with different introgression that fully coverage the genome of the Pinella allele. So it allows us to based on this set, to perform a preliminary experiment with small groups of beels, and maps area that we suspect that affect some phenotype. And in the next stage, we can select some recombinant lines in a specific region that we suspect would affect the phenotype from the large population of the 1,500 plants that cover by the introgression the suspect area, and really shrink the interval to detect some genes that affect the phenotype. So this set, this 53 set in this day is in seed increase. We want to produce a lot of seeds from each build, and also we create an F1 to the Leah to anyone who want to examine the heterozygote state in his experiments can do it also. So let's demonstrate some analysis in the R. So first, before we upload the data must be in an Excel file, in the Excel file, save as a comma delimited format. This is the format that R can read in the RQTL package from Carl Bormann. So the file must be built like this. So the first line is the genotype, the name of the genotype that we have. And in the second, in the other columns until here, we have the phenotypes that I measure. So it's Bill hybrid bricks, Bill's bricks, Bill's total yield, and we measure also a Tobamo brown rugose fruit virus. That I believe that anyone who works with tomato familiar with this virus. So we're doing I'm going to show you the analysis on this phenotype. So as I mentioned, this is the name, and until here is the phenotype that I measure on this population. This is the 53 set of the bills. The two rows here must be clear from anything, and from here it's the genotypic data that I have. So in the first rows, we have the marker's name that contain the physical position and the chromosome, and this is the name of the marker. In the second row, we have the chromosome number 1 until 12. There is 12 chromosome in tomato. And in the third row, we have the genetic distance. This is the centimorgan. So as you see, there is some markers that, like here, this is 3 markers that have the same centimorgan, the same genetic distance, because they don't have recombination between each other. Okay, so let's go to the R. So the R is a very friendly software. There is a lot of data in the internet that you can use for, especially for Qtl mapping and the other analysis that others show before me in their presentation. So let's upload the programs that I want to use it. It's called Qtl from Carl Bormann. So let's upload this program. So the first step for Qtl analysis is to upload the data, the read cross. It's called read cross command. Upload the Excel file that I show you that contain the genotypic and the phenotypic data. So I upload this data. And as you see here, I have in some information. So we have 53 individuals, 53 lines, 7,699 markers, and 16 phenotypes. The cross type, he recognizes that is a backcross population. But I have a warning message that some markers at the same position as I showed you before have the same centimorgan, have the same name. So what I want to do is to jitter map, is to create a linkage group between these markers. So I ran this command, and as you see here, I have the data on this population, the number of chromosomes, number of phenotypes, the percentage of the phenotypes for each phenotype. total markers, the number of markers in each chromosome, and here I have the genotype present. So AA indicates the cultivated allele, the Lea allele. So I have 90.5% from the Lea allele, and it's reliable because it's backcross 2 self 6. And I have a 9.5% from the Pinelli allele in all the genome. Okay, so the next stage is creating the linkage map by calculating the genotype probability. And from this stage, I can start to doing the Qtl analysis. First, if we want, you can show the genetic map. So you can see that the genetic map is very tightly. There is the markers is very closely to each other, both physically and genetically by Santi Morgan. Okay, so let's do some Qtl analysis. Okay, so as I mentioned before, I'm going to do the analysis on the 53 set on the tomato brown rugose fruit virus phenotype. So what we do, we grow the 53 set in the greenhouse. We inoculate the plants, and we phenotype them by ELISA test. It's a quantitatively ELISA, and we suspicious in some resistance for the tobacco for the virus from the Pinelli accession. So let's run the analysis. And let's plot the Manhattan plot. Okay. So in the x-axis, you can see the chromosome from the tomato, 12 chromosome, and in the y-axis the significantly of the markers. So as you see here, there is a very significant area in chromosome 2 that affect on the tobacco resistance, but I want to I want to see if there is the effect of the Qtl. So okay. I forgot to run a command of the Qtl chart, but now I can see the effect of the Qtl of the markers. Okay, so chromosome 2, if I click on this significant marker, I can see is a Anova test. As you see here, 1 is suspectable, close to 1 is suspectable, and close to 0 is resistant. So lines that have the AA, the cultivated allele in this region, are susceptible to the virus, but lines that have the Pinelli allele, the wild species allele, are close to 0. They're pretty resistant to this virus. Okay, so as I mentioned before, I have a Qtl. I have a mapping, an interval an interval for this phenotype, an interval on the genotype that affect the phenotype. And I want to test in a large scale of a recombinant in this region, in chromosome 2, at about 37 million base pairs, to shrink this interval and to detect a specific gene. So if I go back to the presentation, we can show here that I chose a large set of recombinant lines from the population that have a recombinant in the region of the interval. And as you see here, this is the ELISA scores for each line. What's marked in green, it's a resistance, and in red, it's susceptible to the virus through the ELISA test. As you see here, there is a combinant, there is a two recombinants that marked in orange in this region of the TM1 gene. So I shrink the interval to about 10 kb that included the TM1 gene that affect the phenotype, the resistance to the tomato brown rugose fruit virus. So the TM1 gene is a gene that we suspect that affect the phenotype that gave the tomato the resistance to the virus. Okay, thank you very much for listening, and I hope I help you to understand deeply how to use our software. And also you can use this population. This set is in Italy this day, UK, and the Netherlands. And you can use this population for mapping your data. And after you suspicious some region that affects the phenotype, we can send you a large amount of recombinants of your suspected region. And then you can continue for some validation and to detect some genes. Thank you very much.
Thank you, Shai. Very interesting presentation, and also nice to see our tutorial in this workshop as well. So I hope it was interesting for the audience, and I will leave questions to participants. I see a hand raised by Ian. So Ian, the floor is yours.
Hi, Shai.
Hi.
Yeah, great. Very nice overview of the things, and a very practical one, I must say. Very nice. I'm just wondering about, just curious about the last slide with A62 and A64. Can you explain the genotype of these two guys? I mean, obviously you have islands of 3 within 1. How can you explain such a genotype? I mean,
A62
and A64.
Yeah.
So
the combination between this between the markers, it's a 3-1-3-1. This is your question?
yeah, that's my question actually.
Yeah.
So
okay.
So this is in what distance is it? I mean, what's the distance between the markers? Because obviously you have like a research, like a zipper here between.
Yeah. Okay, before I doing the analysis, I filtered the genotypic data by Tassel and by R for markers that are not informative or their deep sequencing is not well. But I have some markers that I that I used to see like that performing like this, but I designed some markers between these markers. As you see, all the blue ones that there are markers that I designed. So you can see that the marker that I designed actually gave us the true genotype. So so I believe.
Yeah, you know why it could happen. What you see here might be polymorphism you see in the wild, that then when you decide whether it's 3 or 1, it could be that your wild species is heterozygous, so it does give you the 2 alleles. So, I mean, as an error, you assign it to 3, but it's probably 1. Because I find it very hard to believe that the true haplotype here is 1 3 1 3 1 3 1 3. I mean, that's really strange.
Actually, I have this situation just in the top population, in the indeterminate population, but in the Lea population, I don't see this situation. So yes, I believe that it's an heterozygote, but also, as I mentioned, I designed a marker for this region and to detect truly genotype.
Yeah, I can tell you that in the case of the champ, when Shevach does, when he extracts the genotype, he did some genotype, a brief one for some of it, and if you don't do it within each family, then you end up with things like that. So you do get kind of a zipper of the genotype. So, but in your case, you have only 2 parents, so it's the only reason I can think about that is that you have an heterozygous donor or something. Yeah, okay.
Is there any contribution of structural variation that might come from, because I see a lot of also NAs in some of the markers. So I wonder, where do these missed call come from? Is it the marker which is not working, or
it's markers that don't work, and in our lab we don't like to use the imputation because we don't like this. We don't like this, to use this analysis, the imputation. But
yeah, no, I agree.
But yeah, I believe the NA that it's because the SPET analysis, the SPET genotyping it's not work here. Yeah.
Yeah, what I'm saying is that could also be to the fact that the site, the restriction site where the SPET marker is developed is not present. So it might be actually or due to either to structural variation or to polymorphism in the enzyme cut site. So, but I agree with you. It's this is not probably the best thing to do imputation here. I see there's a question from Leah Rosenthal. The question is, the top population has 3 parents, isn't it? Maybe Leah wants to take the floor, or you can reply to the chat.
Yes, yes. As we see here, the F1 from the cross between the Pinelli and the Lea crossed to the top. And from here we continue to backcross. So this population contains 3 alleles, yes.
That's very interesting. Is there anybody else would write his response to Yael remark? Yes, but I hope, Leah, you don't have the mic, but I hope that the answer from Shai was satisfying, which I think it was. Is there any other question from the audience? Perhaps people doing experiments on tomato? I think that already preliminary data is available, so I wonder whether the Wageningen team or the Essex team could probably chime in and tell us how it's going.
Sorry, what? Sorry.
No, I was calling for the audience to make questions to you, if any. And but I don't see volunteers. So in the meantime, I have a question for you. And actually, the question is, what about the selection of the wild parental? I think this is a very clever design and a very nice design. But can you tell us? Can you elaborate something about the specific choice of that specific Solanum Penelli accession, and whether you are aware of other wild relatives would be available to build similar population if it wasn't done already?
Yes, of course. So in our lab, we are experts in create a population with a wild species. So population in this scale that contains 1,000 lines, we don't have. This is the uniquely of this population. But we have another population with another wild species, for example, the other accession of Pinelli, the LA716, we have a bill population, and also we have the IL's populations, the famous IL population that contains 70 lines, and we have a Neolithic population bills and Chilense population bills, and every year we doing crosses with another wild species to create more population, because it's a really useful tool for research and for breeding also, because when you have a complete ready population of bills, you can grow them and you have cells from them, and you get the same genotype that you get from each line. And you can continue if you have some newly, for example, new disease in the market, you can screen all the population and found the resistance in the bills population or in the wild type itself. So it's a really nice tool and really useful.
It is. Thank you. So I see a question from Luisa Dever. Is this for 3 parents? Is there a limit on for how many parents you can include in the analysis?
Yes. For this specific Qtl analysis, the package is just for biparental population that contains just 2 alleles. Yes, but when I prepare the data of the top population that contains 3 alleles, I filter out the third allele. Okay.
Yeah, because the challenge then is in reconstructing the haplotypes. And so actually also to chime in and to answer also to Lisa, the framework that Shai is using, which is RQTL, it's actually, it has the capacity to deal with more allele. And indeed, it's the same framework that we are using for the mapping of the magic population. But of course, it's actually designed in a more flexible way. RQTL was the previous version, and now it's developed in a more flexible way that allows different contribution of different parents. So there is some flexibility there, but it's true that it becomes rapidly very challenging as you increase the number of haplotypes, especially when you deal with crosses which are not balanced or with a complex pedigree like the one that Shai is bringing.
As Leonardo showed before me, that it used RQTL2 for the magic population. So it can be used for another population, but this specific package, the RQTL1 package, is specifically for biparental population.
Yes, indeed. Okay, thank you for your answer and thank you for the question, Luisa. Are there any other questions from the room? the virtual room. So if not, maybe we can wrap up the morning. I hope it was interesting for you. And Shai already made a remark about the breeding potential of this population. So I will ask the other speakers to say farewell to us by telling something about what is the way ahead in their population, and especially the way ahead for in a breeding perspective. So I will give the floor to Leonardo for a very quick comment on this.
Yes, thanks, Matteo. So yes, actually the way forward, we may apply different strategies, as I was mentioning at the end of the presentation. Of course, the production of hybrids, maybe with material coming from breeding programs, may be a good way forward. After we, of course, identify Qtls we think are carrying solid candidates. But I see as most powerful tool maybe using genome editing to manipulate actually our genes of interest and maybe produce materials that can afterwards be used in producing pre-breeding populations. This is what I think it's the most are the most promising approaches.
Thanks. So I was saying to Manas and Shevach if they want to chime in and tell us something about the breeding perspective of their work and what lays ahead. Well, the perspective in terms of breeding potential in those populations are pretty clear, and we all look forward to the innovation that we can provide. So for the work package 1 members still online, next deadline we have in our work package, it's deliverable 1.4, 2 years into the project, in which we will include the preliminary results that you showed today. Yes, we can hear you. Can you leave us with a 1-minute impression about what is the breeding potential of your work and what is the way ahead?
Okay, so the way ahead basically is after this year we we performed the propagation. It will give us enough seeds to verify the phenotype in multiple environments. So next year it will grow. This this population will grow in at least 2 to 3 environments in Israel. It will also grow in 1 to 2 environments with our collaborator in California. So this will give us also a range of useful phenotypes, field phenotypes. Yes, and probably some things I'm just not thinking of right now, but we of course also developing already nils for some Qtls we think that are relevant. We want also on the molecular level to validate them. Yeah, I think it will really depend on the data we will get on the beginning. You know, when once we get the genotype, it will maybe guide us a bit more into which direction we will dive into more.
Great, thanks Shevach for this perspective. And once again, thank you to all the speakers. Thank you to Christina, who very gracefully managed the call, and thank you for all the attendees. And I hope to see you in the future and discuss further about the impact and progress of CAPITALISE and other things. Thanks.